Good morning. Let's discuss about mitosis. Mitosis is the process of cell division. Mitosis. Mitosis is the process of cell division where a parent cell divides into two daughter cells. Those two daughter cells will be having the same number of chromosomes whatever the parent has. That's why it is called mitosis. It's also called equivaginal division. Mitosis is also called equivaginal division because it divides equally. Okay. The mitosis word derived from a Greek language. The word comes from Greek language. Let us now dive into the introduction. Mitos. Mitos means thread. Mitos means thread. Oosis means state. When the cell looked through a microscope, it looked like thread-like state. That's why they named it mitosis. What is this thread and who is this thread? Is this thread a chromosome? It is not a chromosome, but it is a spindle fiber. Spindle fiber. Who introduced or who explained this term? Strasburger. Strasburger. He explained this term in plants. And another man, Fleming. Fleming explained mitosis in animal cells. Animal cells. So Strasburger explained the mitosis in plants. Mr. Fleming explained mitosis in animal cells. Okay. Where it takes place? Where do mitosis takes place? Where do mitosis takes place? It takes place in somatic cells. Somatic cells and also in germ cells. Somatic cells are body cells, germ cells are reproductive cells. Somatic cells such as plants, lower plants like algae and bryophytes, they have somatic cells, body cells. Their body divide and produce new organisms, whereas higher plants like sporobiotic plants and humans have body cells. Okay. Mitosis happens in both the conditions. If the question is asked where do mitosis takes place, whether haploid condition or diploid condition, the answer is mitosis takes place in both the condition, haploid as well as diploid. And diploid. Haploid found in plants like algae and bryophytes, their body divides and produce another organisms. Diploid like human body cells. Okay, 46 number of chromosomes, where haploid only 23 number of pairs of chromosomes. Right. Germ cells, as you know, it's a reproductive cells, sperm and egg. And the another question if asked like this: where do mitosis takes place? The germ cells are in reproductive cell, germ cell or somatic cell? The answer is mitosis takes place in both the places, like germ cells as well as in somatic cells. But the thing is, during germ cells, mitosis takes place in multiplication phase. So that is a small difference. In germ cells, during multiplication phase, mitosis takes place. Don't forget. Mitosis is also called as equational division. Equational division. Why it is called equational division? This is a parent cell and the parent cell divides into two daughter cells. This is 2 in number. 2 in number stands for 46 chromosomes, right? 46 chromosomes. When the parent cell divides into two daughter cells, the parent cells, two daughter cells also carries the same number of chromosomes, equally, dividing equally. That's why it is called equational division. So mitosis, another name is called equational division. Okay. Steps involved in this mitosis. There are 2 steps involved in this mitosis. What are those steps? Or stages of mitosis. What are the stages of mitosis? There are 2 stages. The first stage is karyokinesis. The second stage is cytokinesis. What do you mean by karyokinesis? What do you mean by cytokinesis? Karyokinesis means karyon, division of nucleus. Karyon means division of nucleus. Karyon means nucleus. Kinesis means division. Division of nucleus. Cytokinesis means division of cytoplasm. Cytoplasm. So, karyokinesis means division of nucleus. Cytokinesis means division of cytoplasm. The karyokinesis has 4 subdivisions. 4 phases. What are those phases? Prophase, metaphase, anaphase, telophase. There are 4 phases in this mitosis. Let us discuss about one by one. Before discussing a prophase, let us understand interphase. Interphase. Interphase is one of the phases in the cell cycle, in the mitosis, where cell prepares himself for the cell divisions. It prepares physically, right? Like preparations like cell growth, DNA replications, growing in size, right? All these are getting ready for the preparation of cell divisions. That's what happening in the interphase. In the interphase, basically, interphase in S phase basically divides replication of DNA. DNA replicates in number. Let's see how it replicates. We have here centrioles, right? This is called centriole, and we have nucleus. Nucleus has nuclear membrane, nuclear pore, and this is centriole, and this is centriole. Okay, and inside we can see number of chromatin fibers or else spindle fibers. So, centriole started dividing in this stage itself. Centriole start dividing What happens here now? The centriole started dividing. Why it divides? Because after dividing, the centriole has to move to opposite poles. This centriole will be broken in two pieces, and one piece will come here, the other pieces will be there. That is the purpose of a centriole division. Centriole will divide and move to opposite poles. Started moving. And we have chromosome fiber. Chromosome fibers or spindle fiber, and centriole and well-defined nuclear membrane, all these are there. And also in this stage you need to remember is chromosome-like structure formed. Chromosome-like structure formed due to condensation of spindle fiber. The spindle fiber, what you see inside, they condense themselves, they join together and makes trying to make chromosomes. That's what we need to understand here. Chromosome-like structure formed due to condensation of spindle fiber. That means it is not exactly chromosome, right? It's going to be the chromosome. Next phase is prophase. Did I tell that DNA replicates here? Yeah, the main point is DNA replicates. How it replicates? For example, this is a chromosome, correct? And the chromosome has two pairs of DNA. These two pairs of DNA, what happens? It replicates. Already I told you the somatic cells all have 46 number of chromosomes. There is no change in this phase. 46 number of chromosomes remains same. But DNA, only the DNA replicates. This is a chromosome, and there's a two pairs of DNA within the chromosome. That DNA alone replicates. How it replicates? It replicates like four pairs. Previously only two pairs of DNA. After the replication it becomes four pairs. Like 2C value 2 and 4C value. It converted into 2C value to 4C value. How it converted? What do you mean by 2C value? 2C, yeah, C stands for the quantity of DNA, correct? And C stands for quantity of DNA which is present in the chromosome. That's why it is called DNA replicates in this interphase. The next phase is prophase. The prophase divided into two phases. What are those phases? Such as early prophase and late prophase. Early prophase. This early prophase, let us discuss what happens into the cell. We have centriole here. As I told you, centriole started dividing the interphase itself and started moving to opposite. Already, the centrioles moved to opposite in the early prophase. Then let us think about the nuclear membrane. Let us see what is the difference between interphase and early prophase of nuclear membrane. Here, well-defined nuclear membrane is disappeared. Well-defined nuclear membrane is disappeared, trying to disappear. Instead of that, there is a 4 chromosome structure is formed. In this case, the nuclear membrane, nucleolus, and other inner organelles like endoplasmic reticulum, mitochondria, Golgi apparatus, all these inner organelles, nuclear membrane got disappeared in this phase, right? And centrioles started moving to opposite poles. See, already centrioles started moving to opposite poles. The nuclear membrane, which is present here, started disassociate. Nuclear membrane started dissociate. That's what we should remember here. So nuclear membrane, nucleolus, and all this disappear, dissociate. Okay, centrioles and come to opposite poles. And somewhat the 4 chromosome is formed. And let us now move to late prophase. In this late prophase, we have a centrioles in the opposite poles, and we have a spindle fiber, right? Spindle fibers, 4 spindle fibers scattered here and there. Chromosomes go and attach, actually. Chromosome is going to attach to the centrioles, or chromosomes. And near the centrioles, there is a small finger-like projections are called aster rays. And this is centriole. Centriole and aster rays together called aster. Centrioles and aster together called aster. And we have spindle fiber. Okay, spindle fiber. So centrioles plus aster plus spindle fiber. All 3 together called it as spindle apparatus. Spindle apparatus. In case need point of view, the question is asked, how many spindle apparatus are there in the prophase? The answer is simple, only 1 spindle apparatus. What are those one? How it forms? Centrioles, the centrioles, and then aster rays, or which is very small, like finger-like projection, which is present near the opposite poles. That is called aster rays. The centriole, aster rays together called aster, and then spindle fiber. All these 3 together constitute a spindle apparatus. The spindle apparatus are divided into two things, like one is continuous spindle fiber, discontinuous. First, discontinuous. Discontinuous spindle fiber, or else it can be called chromosomal fiber. I will tell you why it is a chromosomal fiber. And another one is continuous spindle fiber, right? Or else interpolar fiber. This one, or else interpolar fiber. So Let us understand. In plate prophase, the chromosomes, the spindle fiber go and attach to the chromosomes, wherever it is, go and attach to the chromosomes, and thus we have a centrioles and then aster rays. All this together constitutes spindle fibers constitute spindle apparatus. The spindle apparatus are divided into two: discontinuous spindle apparatus, spindle fiber, and continuous spindle fiber. Right? The next phase is metaphase. The next phase is metaphase. What are metaphase? Again, we have a cell, correct? And we have centrioles, and we have long fibers. Continuous, I did not explain about continuous spindle fiber and discontinuous. Okay, let us see here. There are two types of spindle fibers in this late prophase. One is discontinuous, or else it is called chromosomal fiber. See, there are four spindle fibers are inside. 1, 2, 3, 4. These four spindle fibers, one for here it starts here and comes and attaches to a chromosome, and here starts here and it comes and attaches to another chromosome, right? So there is that is called discontinuous chromosomes. Likewise, 1, 2, 3, 4. Whatever the chromosome spindle fiber go and attaches to the chromosome, they are called discontinuous chromosomes. Sorry, discontinuous spindle fiber. The other two spindle fiber, continuous spindle fiber, it starts here and ends in these opposite poles. There is no any cut in between. That's why it is called continuous spindle fiber and discontinuous spindle fiber. Let's come to metaphase. In metaphase, we have a long fiber which starts in opposite pole each other from here to here, this end to this end, and chromosomes come and align on an equatorial plate. Chromosomes come and align on an equatorial plate. Let us see how it comes and align. This chromosome connects, this symbol attaches here, comes here, and then either side. Right? So first point, chromosome align equatorial plates. See, here the chromosomes were scattered here and there, but in metaphase, chromosome come and align on a center part of the cells, in the middle of the cell. It has to arrange. It has to be arranged, and you need to understand how it arranges with the help of spindle fiber. Those spindle fiber or chromosomal fibers helps the chromosome to come to a equatorial plates. How they bring them to the equatorial plates? Due to differential contraction. Due to differential contraction. So chromosome come and align on an equatorial plate due to differential contraction. What is differential contraction? See, therefore, say for example, chromosomes are here, scattered here and there in that phase, okay, the prophase, and it has to come to in a center. For example, there are spindle fibers are here. Those spindle fibers, this spindle fiber has to bring the chromosome to the center. For that, it has to do some work. It has to take some effort. Some chromosomes are near to the equatorial plate, takes little effort to bring. Some chromosomes far away from the equatorial plate takes extra effort to bring to the center. That's why it is called differential contraction. What is contraction? The spindle fiber contracts, flexible by itself, contracts by itself to bring, push and pull make an adjustment, make a construction over there. It's actually doing a construction. That's why it is called due to differential contraction of spindle fiber. Due to differential contraction of spindle fiber, the chromosomes align on equatorial plates. Yeah, and also you can ask me a question. Chromosome here it's only two single chromosomes. Then here how come two chromatides are formed? Okay, it duplicates. This chromosome duplicates itself. How it duplicates? From its body, it divides into another half, and this half is called sister chromatids. Sister chromatids, and this sister chromatids also takes two pairs of DNA. Clear? So this is centromere. Centromere. This is centromere. The next phase we will discuss about how centromeres divides and move to opposite poles. Anaphase. Next phase, anaphase. In this phase, the same chromosomes, right? We have long spindle fiber and then short spindle fibers. Okay, 4 short spindle fibers are present. In this 4 short spindle fibers be shown like this in this phase. The chromosomes are seen like this. See, this is a chromosome. The spindle fiber comes and attaches to the chromosomes. After that, it splits, trying to split the centromere. What is centromere? This is centromere. Due to differential contraction or else due to contraction. Okay, what happens? There is a split in between the centromere. The centromere splits. As a result, the chromosomes after the splitting of centromere move to opposite poles by push and pull movement of the spindle fibers. This spindle fiber just pulls towards the opposite pole. This spindle fiber pull towards the opposite pole. For example, here the centromeres split. Centromere split in the center. And second point, there is a formation of interzonal fiber after splitting. There is a formation of interzonal fiber. So we can write interzonal fiber. Formation of interzonal fiber. What does this interzonal fiber does? Interzonal fiber in the middle, it pushes the chromosome toward the opposite poles, whereas the corresponding spindle fiber pulls the chromosome to the opposite poles. As a result, the movement while the chromosome moves to the opposite pole, now the chromosome appears in different ways, different shapes and structure. And let's see what are the ways and shapes and structure this chromosomes looks like. When it pulls, it's like T shape. That means the spindle fiber pulling the chromosome to opposite poles. When you observe during that time, when you observe this spindle fiber through microscope, it appears like this. One is T shape, another is J shape, The another one is L shape. The last one is K shape. This shape called Zup. This is called L shape called Zup metacentric. This is K shape chromosome called metacentric. This is called telocentric. T shape, telocentric. And this is called L shape called acrocentric. So that is how the chromosome appears during this anaphase. Let's move to another phase called telophase, the last phase. This is where the place cytokinesis takes place. When do cytokinesis takes place? The question asked. The answer is the cytokinesis takes place in animal cells in the beginning of telophase itself. See, this is an animal cell and cytokinesis started. There is a formation of furrow. Furrow formation takes place. The furrow slowly develops and split the cell into two halves. Here. Yeah. And this phase is called reversal phase. Reversal phase. Why it is reversal phase? I'll tell you. Very important. Reversal phase. In the prophase, what did we see? In the prophase, nuclear membrane dissociate, right? Not only nuclear membrane, nucleolus, endoplasmic reticulum, Golgi apparatus, all these organelles nuclear membrane are dissociated, right? Where? In prophase. But here reversal happens. Nuclear membrane, all this nucleolus, all these inner organelles membranes are reappear in this phase. That's why it is called a reversal phase. Right. Another point we need to understand here is chromosome. We saw here in the prophase that chromosome-like structure formed due to condensation of spindle fiber, but here chromosome decondensed to form spindle fiber. Just opposite. Chromosome decondensed to form spindle fiber. In that prophase, spindle fiber condensed to form chromosome. Reversal, just opposite. So therefore the cell furrows develop more and more, and finally it divides into two daughter cells. And the two daughter cells carry the 2 in number, carry the same amount of chromosomes. 46 chromosome, and the daughter cell also carries a 46 number chromosome, and parent cell also has a 46 number chromosome. That is the significance of mitosis. So what is the significance of mitosis? They maintain the same number of chromosome, however it divides. Yeah, that's the significance. We are in the last stage of mitosis. The significance of mitosis. What are the significance? What are the significance of mitosis? It helps in growth and development. It brings new organisms by dividing equally. And then second point, repairs and regenerates. Repair and regenerate. For example, if your plant body, some of the part of the plant body is broken, and that place is replaced within 3 months, some days. And same for us also. When we get wound or something happened to our body, and it regenerates and replaces the new cells. That's why repair and regeneration. Asexual repression and vegetative helps in the asexual reproduction as well as in vegetative, cutting, grafting, etc. Fragmentations, right? And then last point, maintains the same number of chromosomes. The same number of. So finally, the mitosis maintains the same number of chromosomes, however it divides. Thanks a lot.